All right, guys, so today we're going to start discussion about genetic engineering. And when we talk about genetic engineering, there are like 4 really specific things that we're going to talk about. We're going to talk about selective breeding, we're going to talk about hybridization, DNA manipulation, and cloning. And out of those 4, probably the most important one that we're going to talk about is DNA manipulation, okay, because it's probably the most complicated, but it's probably also the one that you guys are most familiar with. So we're going to go ahead and get started. First thing, all right, genetic engineering, let's give you a little definition. This is when you make changes in the DNA code of a living organism. So this differs from mutations because remember, mutations is a change in the DNA. So you notice how there's not really much of a difference, but in genetic engineering, it is humans and other organisms that are making the genetic, it's a purposeful change, okay? So you're making changes for a purpose in the DNA code of a living organism, all right? Next thing, let's talk about selective breeding. So here's your first type. Selective breeding is when you choose the best traits for breeding. Okay, so if you have a dog that lives up north where it's really cold, you want to choose the best traits to give that dog a really thick winter coat. Okay, so that might be like a golden retriever or a Saint Bernard. Okay, dogs like that. All right. Most domestic animals are products of selective breeding, like I was just talking about with the golden retriever, the chihuahuas, all of those different types of things. Okay. So selective breeding is when you choose the best traits for breeding, and these are like domestic animals, so cats, dogs, even horses. Okay. So like Clydesdales. Let me think. Little miniature ponies, all of those, horses, even cows, sheep, and pigs. So guys that do FFA or girls that do FFA, you know, if you live on a farm, you're going to go and you're going to show your cows, your sheep, your pigs, your lambs, all of those things, okay? And you know that when you go there, you want to have the cow or the sheep or the pig that has the best breeding standards, and you get those standards by selective breeding, all right? Hybridization. So here's our next type. Okay, hybridization is when you cross dissimilar individuals. So individuals that are not similar to bring together the best traits of both organisms. Okay, this produces hybrids. Now, hybrids can be different species crossed together, or they can be different types within the same species. So an example that of different species crossed together might be with you cross a lion and a tiger and you get a liger. Okay, that's an example of hybridization. You're crossing 2 different species to get one. An example of when you cross the same species but get different types is like a black lab and a chocolate lab. Okay, they're both types of labs, but they're 2 different types. You've got a brown and a black. Okay, so a brown lab and a black lab are also examples of hybridization. Okay, so when it says that they produce hybrids, it's when you cross 2 dissimilar individuals to bring together the best traits of both of them. Okay, and that produces hybrids. All right, here's 2 examples of hybrids. Okay, so you've got a golden doodle. This is when you cross a golden retriever and a poodle. You get a golden doodle or a puggle. This is when you cross a pit bull and a pug, and you get a puggle. Okay, so those are 2 examples of hybrids. And then here's the example of the puggle. So as you can see at the top, you're crossing a pit bull, okay, which is right there on the left. And then on the right, you're crossing it with a pug. And then the arrow is pointing to that puggle. So that's what you get when you cross those 2. All right, next is inbreeding. Okay, now this is one of the lesser ones that we're going to talk about. This is going to be really quick and really fast. Inbreeding. Okay, this is what you get when you continually breed similar individuals. Okay, so you don't mix at all. So an example of this would be like pure breed dogs. Okay, you only breed Dalmatians with Dalmatians. You only breed pit bulls with pit bulls. That is inbreeding. Okay, now with inbreeding, there is risks associated with it. For instance, this can sometimes increase the breed's susceptibility to disease and deformities. So examples might be golden retrievers. They often have epilepsy. Dalmatians often have deafness, and pit bulls often have skin disorders. So when you keep breeding down that line with only one type of dog, or only one type of cat, or only one type of horse, it can sometimes result in disease and deformities. Okay? All right. And when you do this, increasing variation happens. Okay, so breeders can often increase variation in a population when you induce mutations. All right, so remember, a mutation is any change in DNA, and we talked in the beginning about how that's different. So sometimes you can induce mutations by if you notice, like at the bottom, there was a this is called a Scottish fold cat. And what they noticed was, way back when, they noticed that this cat had these types of ears. These types of ears weren't normal, okay? It was a mutation. So what they did was they bred this cat with these ears with another cat with these ears, and that's how you get this type of cat. So they introduced these mutations. Instead of not breeding that cat, they chose to breed it. So they introduced those mutations into that breed. All right, mutations can often produce new kinds of bacteria. So an example of this would be oil-eating bacteria. This was especially helpful when the Gulf oil spill happened. It was so awful. Okay, they used these mutated bacteria to eat the oil off the top of the water. Now, granted, it was too much oil for them to eat, but it helped out a lot. Mutations also produce new kinds of plants. So like daylilies, if you look in the picture, that is a daylily. And bananas are actually a result of a mutated plant, and citrus fruits are also results of mutated plants. Okay, so those are just some examples of that. All right, now, DNA manipulation. All right, now comes the time when you really, really, really need to pay attention, because this can get complicated if you don't. All right. This is when genes from one organism are transferred into the DNA of another organism. Now, I know y'all are thinking, what in the world is she talking about? That is so not possible. Okay? Yes, it is, and I'm about to show you how. All right, so remember when we're talking about this, that this is when you take genes from one organism and you put it into another organism. Now, oftentimes, we use bacteria. Okay, so we'll take animal DNA or human genes or animal genes and put them into bacterial DNA. And we're going to talk about why in just a second. But let's see how this happens. All right, so you have your diagram that I gave to you at the beginning of class. So now is the time when you need to start labeling it. Okay, so as you can see, move my cursor over here. We have a human cell, right? Okay. And we have a bacterial cell. Okay? Human cell, bacterial cell. Or you can call this an animal cell. Okay? Animal cell, bacterial cell. Now, you notice that there's a difference here. If you look up here, this human cell, it has a nucleus. This bacterial cell does not. That was something that we learned about. Prokaryotes, pro equals no. Prokaryotes have no nucleus. So that's one of the differences. All right? Nucleus, no nucleus. All right, down here you can also see this is the bacteria's DNA. Notice how it's in a circle. If you look up here, this human DNA is not in a circle. It's all jammed up into this tiny nucleus right here inside the cell. This bacterial DNA is not like that. It's in a circle and it's free floating inside the cell. Okay, now, because of this, bacterial DNA is circular, okay, and it's known as a plasmid. All right, so remember that. One of the most important words you will learn this unit. Bacterial DNA is known as a plasmid. It's in a circle and it's in this cell. Human is not the same way, okay? Now, first step, you need to take out the DNA of both of these things. So as you can see, we have this arrow and it's showing you how you're taking out this DNA. All right, same thing here. You're taking out this bacteria's DNA. Next thing you need to do is you need to figure out a way to get these genes out of this DNA. Remember, you're taking genes out of one organism and you're putting it into another. So in this case, we're going to be taking the genes out of the human DNA. All right, now as you can see here, we've got this piece of gene. Okay, here's your gene. this gene or this section of human DNA is cut out, and we cut it out using restriction enzymes. So it's like these scissors, okay? You can think about restriction enzymes as scissors. They come in and they cut the human DNA and they cut out that gene, all right? Down here, you don't need to cut out a gene. All you need to do is cut the bacterial DNA so that we can place this gene into this DNA. So all you're doing here is cutting it, and you're cutting it using the same restriction enzymes, okay, exact same ones. Now what you got to do is you have to take this gene and put it into this DNA. And when you do that, you make this new type of DNA, and it's called recombinant DNA. So you're recombining things, okay? Makes sense? You're taking one thing and you're combining it with another. That's a way to remember it. Recombining DNA, okay? You're taking one DNA and you're combining it with another. Recombining DNA. Recombinant DNA. So this is when you take the gene from the human DNA, right here, all right, and you place it into the bacterial DNA right here, and you get this type of DNA called recombinant DNA. Now, after you do that, the only thing you have to do is place it back into the bacterial cell. That's it. So what they do is they take this DNA with this newly inserted human gene, and they put it into this bacterial cell. Put it back into the main bacterial cell over here. Just put it back in there. And then they just let the bacteria divide. Now, because bacteria divide so rapidly, okay, they're able to produce a lot of copies of this main gene that you need. This is often used with human insulin. So they'll take the insulin gene out of the human DNA, they'll put it into this bacterial DNA, and all of these bacteria can start making this new gene for insulin. Okay, so that way you can give it to diabetics, and hopefully this can be used as a cure for diabetes. All right, moving on. Next thing, why do you use bacteria? I just explained this to you. Bacterial cells are often used during genetic engineering because they reproduce quickly and enable more copies of the newly inserted gene to be made. Humans take about a year to reproduce. Remember, it's about 9 months to a year. So you have to wait for 9 months to make this gene. That's not going to happen here. Bacterial cells reproduce every 20 minutes. They double in size. They double in their population. So that means in 20 minutes, if you had 100 bacterial cells, you're now going to have 200. Okay? So they reproduce a lot more quickly. That's the reason that we use them. All right, DNA manipulation can often reproduce transgenic organisms. So this is when you have organisms that contain genes from other species. Okay, so you've got like this. So microorganisms from bacteria. You might have bacteria that contain genes from other species. Mice, animals. This is a big one right here. They often insert genes into mice for medical uses, and I'll show you an example of that in just a minute. And then plants for agricultural uses. So let's move on and I'll show you some pictures. All right. So this is a transgenic organism. This is a mouse. So they've taken the gene for glowing out of a jellyfish, and they put it into this mouse. And now these mice can glow in the dark. All right, same here. They've taken cells out of the human ear and implanted it into this mouse to see if this mouse could make a human ear. And they did. Here it is. Same thing right here. They're just showing you how the mouse can now glow in the dark after they've taken the genes out of it and put it in there. All right, now with cloning, this is another big one. This is when you have a member of the population that is genetically identical to something else. Okay? It is very easy to clone single-celled organisms. Bacteria are genetically identical to each other. They're essentially clones. They're also single-celled organisms, so it's not that bad. Multicellular organisms are much more difficult to clone, okay? Because there's a lot more cells, there's a lot more genetic diversity. However, there are such things as natural clones, okay? Twins are natural clones. Identical twins are natural clones, okay? And then the first mammal that was cloned was actually Dolly the sheep, and she was cloned in 1997. All right, moving on. Clones do naturally exist. These are known as identical twins. We already learned that. We learned it yesterday. There's Dolly the sheep. So she was the first mammal to be cloned. And then these two diagrams really show you how to clone, show you how to clone a sheep. Okay? So you take the body cell from sheep A, and then take the egg cell from sheep B. You take the nucleus out, you put the DNA in here, and then that makes your clone. And that is pretty much it.